Hello everyone. Today I will introduce you a software made in my laboratory. It's called GENPPI. It's something like a network interaction generator. GENPPI is a software written in Common Lisp and compiled with SBCL to create complex networks of predicted proteins from tens or hundreds of genomes. Your main limitation is the amount of RAM in your server. For instance, using a conventional computer of 8 gigabytes of RAM, the software can deal with at least 15 genomes, each processing on average 2,200 proteins, Corynebacterium proteins. However, this same machine cannot process 80 genomes with an average of 5,000 proteins from Escherichia coli, for instance. To achieve this last task well done, we need to compile GenPy to use at least 8 gigabytes of RAM and use the program version capable of storing the bulk data on disk instead on memory, the GenPyDB executable. For now, I had compiled all versions available in the binaries folders to use different sizes of RAM. You can check it out in the GitHub. While I do not release the source code, if you need a version compiled with more RAM, please get in touch with me. SBCL includes the whole core of libraries for each executable, and it explains the size of the software in megabytes. Till today, I have not a graphical interface for GenPy since I conceived it as a command line tool for the Linux operational system. Calling the software without any arguments results in printing all possible parameters in the combinations. It also happens when starting the program with the -help option. I will show you a fast track to obtain results with the software. First of all, we need to create a folder containing multifasta files. Here, I will show you each folder from a species of bacteria. The example I will show you is the Bacteria apicula. In this directory, I have only 5 genomes of Bacteria apicula. The genomes are represented by the proteins. For example, here I have only the proteins from each genome. In this folder, I have 600 proteins on average for each genome. Bacteria apicula is one of the smallest genomes in the world, so it's simple. Because of that, it will very fast for GenPy to produce network interactions from these genomes. We already have a folder containing multifasta files of our proteins. Each file will be treated like a unique genome. I recommend that you name these files significantly because a lot of reports will mention these names. I also suggest keeping the protein names as simple as possible and short. Here you can see that I have a very short name, no special characters to complicate our lives. I have another program that you can use for this test. the task to simplify the name of the proteins. I call it value faster. Value faster tries to figure out a combination of strings identifying without redundancy all proteins with a multifactor file. If such an assignment does not succeed, ValueFaster creates a numerical and sequential identification for each protein and uses it instead of the original proteins. The files that I'm showing to you now were processed by ValueFaster. ValueFaster is also available to download with the GENPPI folder in the GitHub. To run the program, there are a lot of parameters and combination of these parameters. I recommend you to start simple. Probably you will not reach out a comprehensive interaction network on your first try. As you master the simple, do further steps. As a rule of thumb, a good interaction network should represent the majority of your proteins, number of vertices, possess at least 2,000 or 3,000 interactions, number of edges, and on average a few hundred, smaller than 200 interactions per protein. Some proteins can have thousands of connections, but this should not be in a representative number. A configuration that I use too frequently is this one that I will show you right now. The first parameter that I have is pp-complete. Pp-complete is an excellent parameter to run at least once for each set of genomes. It does not restrict the number of interactions concerning phylogenetic profiles, or PP. It just let it go. Depending on a small number of genomes in your folder, the consequence is a massive and undesirable number of edges as a final result. The reason is that for a few genomes very closely related, the majority of the PPs will be very conserved. Try to analyze related genomes, but not necessarily very similar, to diminish the number of possible expected interactions. The phylogenetic profile report only is created when we settle this parameter. If you decide to restrict the number of connections, do not use this parameter, but limit the interactions. Optional parameters to limit edges are pp iter limit, trim, and threshold, just citing some of them. Check out the -help options for a full list of options instead pp-complete. Well, the second one is the x-fixed. It sets the program for comparing neighborhood genes at most -W1 genes and using -CW1 as the smaller number of genes for concluding a conserved neighborhood, or CN. CN and PP are the primary methods to map significant connections in an interaction network. The counterpart of xp-fixed is the xp-dynamic, for example, with the parameter -WS3. It conducts a systematic expansion of windows limits on analyzing a CN. However, dynamic growth has time consuming proportional to the WS value. Another parameter is pp diff tolerated. It gives you more interaction as a final result of the pp analysis. This parameter integers value defines the level of tolerance to accept two PPs as similar between genomes. Another parameter is pp histofilter. It sets the GENPPI to be too restrictive when deciding the similarity between two proteins, but only during the PP analysis. The lack of this parameter can also create a much larger number of edges at the final interaction network. Well, people, let me warn you. This is a very simple example. Bucknera apicula is a very small genome, one of the smallest. So, this execution of GENPPI will be very fast. Let's see that. Ooh, it's done. Well, very fast, don't you think? What happened here? In a few seconds, we got the whole pangenome, including accessory and core genome, mapped, and after that, we analyze the 5 genomes according to the gene neighborhood conservation and the phylogenetic profiles report. The final step was the PPI report generation for each genome. And in the result folder, we have a few reports. For example, we have the gene neighborhood conservation report, the pangenome, the phylogenetic profiles report, all the PPI files, and the PPI report. Let's see the PPI report. Well, for the genome named PA_AQ, we have just 300 edges according to CN and 42 edges according to PP, and so on. The biggest number of edges was mapped for this genome. Let's see that. Now, I will open the biggest PPI network with the program GENPPI. In this interaction network for this specific genome, we have 120 vertices or proteins with 396 Fs. Okay, let's open that. Now I will apply a layout for the distribution of vertices and edges. Okay. Now I will calculate, for example, the average degree for this network. This is the report. And the reading centrality. When we ask GENPPI to calculate the reading centrality, we also make the calculation of other metrics. For example, the between the centrality, reading coefficient, and the reading centrality. I can ask GENPPI, for example, to rank these nodes according to the reading centrality mission. Let's see that. So now we have the top node according to reading centrality, dark green color. For an initial explanation about our software GENPPI, I think that's enough. Please access the GitHub and look for GENPPI. There you will find all the binaries necessary to execute our software, and also some tutorials, including tutorials for the operational system Windows. If you need some adjustment in the binaries, please contact me. For larger genomes, you can have much more time to process all the genomes. For example, considering 15 genomes of Corynebacterium, we have more or less 50 minutes to process all the genomes. But for other genomes larger, like Escherichia coli, you can spend at least 5 hours to complete all the network interactions. Another possibility that we can do with our software, GENPPI, is to generate graphs for a lot of reports created by our program. For example, all reports named report.txt can be uploaded to our server to generate graphical outputs. Let's see, for example, one of those reports. Phylogenetic profile report. Submit the query. It should be fast because it's a very small report. After that, we have two graphs. Let's open that. Well, it's a very simple box plot set because we have few genomes. But looking to these box plots, we can see the median of the number of phylogenetic profiles conserved among the all genomes analyzed. Such a graph can give you a clue about how evolutionary related are each species under analysis. Sometimes we can even separate biovars from bacteria using this graph. All the three reports generated by our program can be used with this web page. For example, the PPI report gives us two other graphs. This graph is the number of conserved neighborhoods that were found in each genome. Here we have the number of phylogenetic profiles conserved for each genome. Once more, this genome has the smallest number of phylogenetic profiles concerning this set of genomes, and this is the most conserved according to the set of all possible phylogenetic profiles between these 5 genomes. For now, that's all, folks. Thank you for your attention. Please use our software. Using our software, you have the power to decide how many genomes of which species you will use to answer your scientific question. Thank you very much.